Welcome to NewYorkBIO's virtual breakfast series, a digital program started in 2020, bringing you fireside chats with leaders from across the healthcare spectrum. This week's guest is Dr. Premsrirut, the founder and CEO of Miramis.
All right. Good morning, everyone. My name is Jennifer Hawks-Bland, and I am the CEO at New York Bio. We're thrilled you're here with us today. Today marks the end of TriNet's sponsorship of our virtual breakfast series. We have been so thankful to have amazing partners like TriNet to sponsor our breakfast series as we've all shifted and done things that we didn't think we could do virtually. We have managed to do it by having great guests every Tuesday morning. And today is no different. I do want to add a little teaser. So next week, Derek and I actually debated whether we should have a virtual breakfast series on election day. But we ultimately decided what better to talk about on election day than politics. So next week we have Mike Mattoon, who leads BIO's Federal Government Affairs Division, and he will be here to prognosticate, predict, and debate. So make sure you join us for that one. It's sure to be lively. But in the meantime, let's continue to talk about science. So I will turn it over to Derek to introduce our guest, Dr. Premsrirut.
All right, good morning. You literally just made me cycle through about 30 jokes that weren't going to work there. So I decided not to interrupt you and not to go there. So Prem, we are super excited to have you on this morning. I love things like this because, we've spent probably a pretty contiguous time within the New York City bio-entrepreneurship ecosystem. And it's always nice to have kind of shared experiences to talk about on this. So for those of you who don't know, Prem is the founder and CEO of Miramis Inc., based in New York City. And Prem, why don't you give a little bit of an intro to what Miramis does, and then we'll talk a little bit about where it all came from.
Hi. So yeah, thank you for having me. It's a pleasure to be here. I think I've been a part of New York Bio for so many years now, and it's nice to be a speaker. So Miramis started about 10 years ago, and we started out of Cold Spring Harbor Laboratory based on using a lot of the technologies that we're developing there, particularly RNA interference technologies. So we had uncovered a way of how to use it in animal models pretty effectively to mimic drug therapy. And so we were able to take that and commercialize that about 10 years ago. And since then, we've been really focused on a lot of genetic engineering and most effectively taking these complex biological processes, learning, building out pipelines to scale them, do them at, high throughput level, and then and make it more cost effective. So this is really our expertise area.
Right. So very basically, you can take a mouse model and make it, closer to a human or closer to the biological pathway that people are trying to study. Ergo, it should be better from a translational standpoint. Yes.
That is one aspect of it. I think when we started doing RNAi, RNAi was really a way to mimic drug therapy. You could turn genes off and on transiently, repeatedly. You could do it in cycles as if you were giving a drug and taking them off and so forth. It was a little bit challenging when CRISPR came out a couple of years later because a lot of companies were pulling money out of the RNAi space and channeling it towards new technologies such as CRISPR. So we had a decision to make what we were going to do, and we really quickly just jumped on it and learned how to use CRISPR more effectively to do similar things that we were doing. So what we were able to do when we put CRISPR into the picture was use CRISPR to make disease, modifying mutations, and then combine it with RNAi to mimic the therapy. So all in one animal, you could, again, make the disease plus mimic therapy. And we started building these preclinical tools for a lot of pharma companies and academic researchers.
Awesome. Okay, so we're going to come back to that, but for now, why don't we rewind all the way back to the beginning? So it's 10 years ago, and you're doing an MD PhD between Stony Brook and Cold Spring Harbor, and where did the idea for all of this come from, and kind of take us back to where you were then when you were thinking about all this.
Oh, I feel a lot younger then, but 10 years does something to you. It's funny because we had received a large grant from the NCI, and they had liked the pipeline that we had developed, and they wanted wanted us to make 1,500 models and have them stored in a repository. So we received a large grant, and this was really my first experience of managing a large project, and I'm still just graduating, finishing up my PhD thesis work. And I was asked, do you want to do this? And I said, okay, let's go for it. So I hired the team, set up the pipeline, reestablished it, and really had to learn how to put a budget together, and it was like, how many of these can you do? How much is it going to cost you? And I'm like, okay, sure. So, but everything, when I finally started to realize, oh my gosh, this media costs this much. Oh, let's scale it down. Let's scale it down. Let's continue to scale it down. What do we really need? What is the minimum amount? And how can we do it quicker? Oh, people cost this much. I need to scale. And
Yeah.
and it was my first lesson in scalability, right? And it really occurred to me, wow, things are cool. Science is cool, but if you can't scale them, there's just no commercial application, right? You got to fit things in a budget. And so that was like, as a just finishing student, my first lesson in this, and building this out. And then after that, the funny thing was I was presenting my PhD thesis. I was doing my thesis defense, and my whole family was there. And I have a really large family, and they're all crazy, and they're all entrepreneurs, and they're all, and and my family at the end of it, they're like, in the public, do you have any questions? And like, my sister's like, I don't understand any of this. What does it mean? And my brother says, well, when are you going to make money out of this? See, and then it was like, I kid you not, a week later, I was approached, by Cold Spring Harbor, by my PIs, by VCs. Hey, that's not a bad idea. Would you want to do it? Put together, an idea, business proposal. And I said, what? Put a business proposal? Okay, sure. I don't really feel like going back to medical school right now. This is kind of fun. So, I did it. And I think I presented, and a couple months later, I had $2 million in a bank and a new company. So that's how it happened.
That is so not how it usually happens. That is just not, that's not the process they use in the books. So I was going to say, if you thought about it, what would you have done differently? But other than, I guess, taking some more time to either think about the idea and plan things out carefully, with that, it really sounds like almost a perfect storm of, really pretty smart people around you laying out a decent case for you rather than you having to sell the case to everybody else.
Well, that's true. And a couple of things I learned. You always need 2.5 times what you think you do. That's number one.
Yep.
Every 6 minutes that you talk to a lawyer is very expensive. That's number two. I don't know.
Someone used to have to keep up with my 0.1s on the other side of that equation. I totally agree.
I learned a lot in that time frame, but it was sort of like, being thrown in a river and learning to swim, out of it. But I don't know if I would have done it differently because, in life, there's choices you make and you just have to go follow through with them. You can't keep second guessing yourself. So, yeah.
Okay, so as you get rolling with the magical $2 million in the bank and all sorts of support, can you talk a little bit about kind of the growth initially? So how did you start with some people? What did your early team look like, and what did your early kind of customers look like? What were some of the things that were probably formational and thinking about, okay, this is really how we have to run this business?
So the lucky thing was I had a nice team of, dream team founders, right? So, they lended a hand in the beginning, and kind of seeded the idea to a lot of people that, here's a service. I think our idea was always to take our expertise and, become a therapeutic company, do things like that. But we had a way to generate revenues early, we didn't need a lot of money to generate the revenues. We already had a working pipeline that we had, funded with, millions of dollars from the NCI. So it was just taking that and expanding upon that, and then building it out. But, a couple years later, it was like I said, you learn, wait, I need 2.5 times what I asked for.
Right.
So, and what we didn't expect was a technology that was obviously going to win the Nobel Prize and, and everything, just to kind of take control of the entire world. And, no one was paying attention to what we were doing anymore. That's the one area where, I didn't anticipate it. And it was really like, now I had a team of 8 people, and, I also learned early people are your biggest assets, right? The right people. But I had this team and I didn't know what to do, but they were all very smart and we were all very interested in making the best system for engineering. So we sort of put the therapeutic aspect aside, and, we decided to go for CRISPR, and we had already been doing it, in the academic space. So it was more so, okay, you can do this. How do you, again, same process. How do you make it efficient? How do you build a process around it to make it so anyone can do it, and that's how we carried on.
Okay, so as you're rolling, eventually you start working with Charles River, which is a household name that everybody knows. So how long did something like that take to put together, and what were kind of some of the major things that it brought to you as a business?
You run into the same people at conferences when we could travel before, so yeah. So
when you could run into people, you ran into people.
Yes,
exactly. So, we were able to, we would see the same people, but I think a part of it was thinking, okay, what is it that we want to do for our business? Where's our expertise, and where are the partnerships? Where are the companies that have their, can help you, right? We ran into a problem building animals at Cold Spring Harbor Labs and then shipping to clients. And it was a good early problem to have, because we realized, wait a second, do we really want to be a vivarium? Do you want to compete with, the really high barrier facilities, like this? No, that's not what we wanted to do. But our clients needed our animals to, either be re-derived or they had to be quarantined for a very long time. And it was slowing down the process and adding to the costs. So again, it was just all about time and costs. So we decided, can we make animals at Charles River? And that way they can just be Charles River, standard, and go into people's, facilities without any quarantine or re-derivation. So we started to subcontract them. And they started to say, wow, this is a big user. They're using a lot of even embryology services that we don't even have. We have to build it out. It was actually a process of us having to teach them what we needed. We had to go there, we had to train their embryologists to be better, to have the efficiency that we did. And it became a real partnership because of that, because we were willing to take the energy that it needed to teach them. You would think this company had all of these things in it, but that's not what they did routinely.
No.
And so, we helped them establish that expertise and that core. And that's why when, in 2015, 16, when they were evaluating, we want to start doing CRISPR engineered animal models, we were the natural, partner for them, because we'd already built it out. We'd already been challenging them to do things of that nature. So they flipped the contract and the partnership. And rather than us subcontracting them, they said, we would like to subcontract you for all of North America.
So basically then you ran their program.
Correct.
Well, that's pretty fantastic. So go ahead, Jennifer.
Oh, I was gonna say, when was that, Prem? 2018?
Yeah, that was sometime, these things, when you work with companies like this, the origin of it started in 2015.
Okay.
But we officially signed the contract 2016.
Okay.
But I can say, it's, and we all talk about this, Charles River and I have been dating for a very long time now. And,
We go and stay.
Yes. The deal happened very quickly. It took 4 straight years of discussions, and then however long it took to get the contract done, very quickly.
Yeah, it was feeling each other out for from 2013 to 2016, and then finally getting engaged 2016.
Well, one of the things that's difficult when you're a larger company that, really has a very set way of doing things in a really straightforward business model is nuance, right? And, in a lot of ways, you have companies where, once they sell the product to somebody else, whatever happens on the back end, they don't necessarily control anymore. So you've kind of taken a place where, people could have been innovative on the back end of things, and you developed this thing where they could kind of capture a little more of that and bring it in-house rather than have it go out and be done all by the end users. So I'd really actually like to talk a little bit about kind of where this sits in, from a discovery standpoint. We have a couple of other things that I think would be fun to get to, but given the fact that, now CRISPR is a tool that people use all over the place, how do you see what you're doing, impacting where new drugs come from, right? Where does the future kind of dovetail in between what we do with animal models and how we think about humans and patients?
This experience has allowed us to work with companies all over the world, and big, from very, very large companies to small academic research labs, and having to understand their goals, their visions, their even how things work from the pharmaceutical side, what they're able to budget, how what are the important factors to them to get something through that clinical pipeline, right? From a preclinical standpoint, what are the endpoints that they need to see? And so all of this knowledge has sort of built up over the years where, now we're building, preclinical oncology pipelines for Roche. We have a number of other pharmaceutical company, partners. We have new models. We understand where their hurdles lie in the preclinical space to have enough proof of concept to take it further. And I think that's where our expertise is understanding, how someone gets it to that IND, how someone, what's the data points they're looking at, and understanding this is not just, about the coolness of the science, but it also about the quality of the data and the timing of it. Timing is everything in pharma world, and if the data is going to take 5 years to generate, it's not worth the time nor energy. So, having to put all of this together and be that arm for a number of companies has been quite exciting and challenging at the same time. But it's allowed us to, it's given us that insight to understand the sort of pre-research work that goes into the clinical development, therapeutic space. So, what it's allowed us to do is sort of stand where we do now, where we're having a lot of, we just signed a new partnership to build out, a potential new therapeutic and design that for for a company. So this is, leading into building preclinical animal models, but now actually building the potential therapeutic itself. So for us, this is a very exciting turning point of Miramis to now, be able to say, yes, we finally accomplished what we always set out to do was, build RNAi therapeutics, and we're heading in that direction now.
And how did you decide to make the shift? Was it natural, or was this something you decided and then went after it?
No, it was very natural. It was, building tools, right? Reagents that you'll use for research. But wait, if this is a tool, can this be a drug? Can we build it a little bit better? Can we look at these, endpoints? We've come far enough. RNAi is particularly, has always been potentially a great therapeutic, right? I mean, some of them have actually been FDA approved now only in the recent years. It's always been a delivery problem. And now that everyone's been working super, on delivery for a decade or more, not only for RNAi, but for CRISPR as well, those things have been able to be, merged and married with the therapeutic itself, right? And coupled. And so now I think it's the time that people are paying attention again to our old friend RNAi, and looking at it as now again, a revolutionary potential therapeutic.
Yeah, well, that's one of the things about tools companies that's really interesting, is that when you're the people that make the tools, you get really good at using them. And you actually get really smart about asking, kind of similar questions that your eventual customers ask, and ultimately usually turn around and say, well, wait, we can do this too. You get questions that you think are really scientifically interesting or relevant from a disease standpoint that may or may not fit into your customer's pipeline questions. But then you say, well, why can't we do this? Right? Because you're ostensibly the best people in the world at using the tool that you developed.
Okay.
Yeah. And it's not terribly hard to look at valuations on, on effective molecules and think about, where they stand in relation to, valuations of effective tool companies. So from a value standpoint, they're pretty valuable assets to have in your arsenal. So why don't we talk about COVID? Because now you also amazingly somehow have a, COVID saliva test that has been, going gangbusters and is now available.
Yeah.
So most of the time when people have a vision with a startup company and they execute on that vision, it's great, and you take it and the vision grows. It's rare that you can also include something else that actually is, tangential yet still works and is somehow also successful. So rewind when did you think about doing this? What did you do? How did you do? I don't even know where to start.
Well, it all started, as you could see the events unfolding right before you, late February, early March, and my closure of my kids' schools. And I'm like, oh my gosh, what am I going to do? So what happened really was, we could see the businesses starting to have to be shut down. SUNY Downstate, which we are located, in the incubator, a block away from the hospital. I'm a volunteer faculty member there, teaching classes there, being, pretty involved. And, we had options, right? Close the company down, go work from home. But, a couple of things that we needed to do. We knew, okay, we need models. We need animal models. We're going to need them. We're going to need to study the therapeutics in them, the vaccines. So that's one thing we did. We raised some money and just said, let's go for it. Let's make these humanized, ACE2 receptor models, both mice, both rats. Let's make some clever ones that, will be important. And let's look, have some foresight into that. And so, we did that knowing that the only available model was cryopreserved, and it was made with really old technology, had, random insertions, 8 copies. And if the world is only using one kind of model, the answers are going to be very skewed, right?
Right.
Models. So that we did. And we are like now actually scaling the breeding of them so that we can finally distribute them. It took some time. And unfortunately, mice, you can do IVF. With rats. No, they're just,
no, huh?
You cannot scale them fast enough. So they're breeding nice and slowly, but the colony's growing. That was the first thing we did. The second thing we did, was really reach out to the hospital. Actually, they were reaching out to the companies. They were short on supply of everything, tips, RNA extractions, kits, and all of these things. They were short on people.
Yeah.
And, I asked my team and said, look, who wants to help? Who wants to volunteer? And everybody raised their hands, and nobody wanted to go home. Nobody wanted to, stay. And I have to say, my 1 week at home, it was just really depressing. I had to get out. So,
Yes.
So it was just a calling, what can we do? Tell us. And it was, we need, when they were going to start this convalescent plasma program. So SUNY Downstate turned into COVID only hospital.
Yeah.
And we started, building out the ELISA assay that Mount Sinai had published. We started, we got the, and you have to realize at this time, none of this existed commercially. You can't just go and buy the protein. You don't have anything at your disposal. You have to make it. Clinicians don't know how to make this stuff.
Yeah.
So, they turned to us, hey, you guys make this stuff, right? Yes, we do. We do this all the time. Sure. So we started to build out these assays, and I got drafted. I had to take my medical degree, my medical training back into action.
Dust it
off and say, okay, let's go. Let's look at the clinical picture of all this. So I joined the convalescent plasma team of SUNY Downstate. And it was, we met once a week, twice a week, and it was gearing up to take the Mayo protocol to then treat very critically ill patients with donor plasmid from recovered patients. But before you do that, you need to measure the titers of these, the serum. and how much antibodies. And so that's what we were doing. We were taking the one we developed. We were taking some of these, rapid ones that were being released from China or here or there, all over the place. People were sending us, hey, you guys have samples. You have a whole bunch of patient samples. Can you test these? Absolutely. We'll test them. And we found a few that worked really well. And so we started being able to measure, antibody titers and look at this. And, what occurred to me, the insight that I saw was, wow, hospitalized patients with severe critical patients have high levels, especially ones that, bind the most specific, regions of the COVID-19, right? And actually, when you develop these assays, you have to realize that, obviously not all antibody tests are made equally, right? Some are made with proteins that have, 90% homology to other coronaviruses. Well, that's going to be very specific, right? You have to look into the details of what you're measuring and understand why you're getting the results you do. So, we started looking at this, we said, wow, wait a second. These patients have such high levels, but the mild asymptomatic patients, which a lot of them weren't being confirmed. They weren't being tested. We had to go around. We made a mobile unit. We had to go around and find these people, who weren't going to the hospital, who weren't, and we were testing them. Wait a second, they have very low levels. What's going on here? And that was the first insight of, and we could see it, antibody levels going up, down, they were not, it was not what we normally would expect. The IgM levels come up 7 to 10 days. The IgG levels follow suit like 14 days, 21 days or so later. It was all over the place. And the news was talking at that time about immunity passport. And I was like, no, not going to work. Absolutely not going to work. And so we had reached out to some foundations and we got funded by the Skoll Foundation to further this research because they were highly intrigued with our findings of, what we were uncovering. And I kept saying, it's got to be something more than this antibodies, it must be a T cell response. It's got to be, just people that don't have severe disease don't need to recruit B cells and make antibodies. There's something going on here. So that was the switch. That was the switch of, wait a second, if you can't tell if someone had it reliably, which we couldn't, we had all the antibody tests at our disposal, and we couldn't do that. So it became, well, if you can't do that, you're going to have to know if you can't tell if someone had it, you're going to have to know if someone has it. And a test is only good for today and maybe tomorrow and the day after, but it's not good for, beyond that. So now we're going to have routine testing. So, okay, how are we going to do that if it costs $150 a pop? That's not possible. I don't care if the government's fronting the bill for every single person. It's not going to be possible. So it was very kind that I had this volunteer, like army of volunteers, postdoc students, and they're like, what can we do? It's like cost analysis. Go find me every single, diagnostic, viral diagnostic test out there and cost it out for me. Give me all the possibilities. At the same time, I reached out to my old colleagues who were building these same assays, diagnostics for their institutions out in Europe. So, in Vienna and Israel and Germany, and they were a month ahead. So they had a little bit more knowledge. And I said, what are you guys doing? And one of them said, oh, ditch the nasal swab, try saliva, it works. It's just as good. We've done it pair by pair in hospitalized patients. And I said, interesting, very interesting. And that was it. That was the aha moment. And I trust his data. I saw the data and I didn't need anything else. All I needed to know was this was going to work. So we started probably towards the end of March, beginning of April, with a pilot project, an IRB study, comparing actually what we would call throat washes. And ironically, it's what Canada instituted, and they said the first saliva test is, they're gargling saline. And we were doing this and it worked beautifully. It was nice. It was a homogeneous solution. It was great. And I thought, okay, how am I going to get this approved? And I had a very intelligent, very smart person said to me, Prem, think wisely. The moment you have to put some kind of solution into someone's mouth, you can never get the cost down. You're going to have that GMP manufactured, sterilized, you're going to be small aliquots. I started looking at how much these saline, what they use insulin shots, or for diabetics who use the saline, like how much of them, I'm like, oh no, I need the cost to be under $10, the whole thing. I need every single thing I use in this. And if this solution is costing me $4, no, it's not going to happen.
Yeah.
So, we pivoted a lot and tested a lot of things, not just, for functionality, but for cost to get the goal was to have the cheapest testing that we could. And I looked across the reagents, looked across everything, and it was okay. We've got to combine samples. We've got a pool. That's it. Most people don't. We can dilute a positive sample 200-fold. There's no reason we can't combine 20, 30, 40, 50 people. If we can do that, we can see that. And that's where we sort of went on this this whole pathway of like building this out. So
if you're combining and pooling samples, how do you know which one is positive?
Right. So that's when they do something called reflexing. So I have to, just take one step back. The beauty of what we were doing, although we weren't publishing any of it, we were talking about it, was almost in parallel. Everything was coming out of Yale. They were publishing exactly what we were seeing in the lab. And it was just like, oh, thank gosh, someone's publishing it. I have, evidence here. Look,
it's validated.
It's validated, but you would not believe the resistance from everyone of the nasopharyngeal swab. I have no idea why everyone has, and some still cling to this idea and notion of poking someone to the back of their brain to scrape out a few cells. Because
it sounds thorough. It literally sounds, it does, right? And you by the description of it, the normal person listens to this and it sounds like this, you're doing something terrible to your face. So it feels like it should be more thorough than spitting into a cup. Right. It's like, well, this is so much easier. How could it possibly be as good as the thing that you shoved into my brain and rubbed around to get all of the stuff that was there?
That's the hard part with scientists, researchers. It's when you're stuck in a box and you think this is a respiratory virus. It has to be here, right? It has to be up here.
Yeah.
And you can't get out of that. But when you just do the studies and you prove it otherwise, it's unreal. I mean, we know now. I mean, Japan just did a 2,000 paired study. 2,000 person paired nasopharyngeal and saliva. Yeah. I mean, the evidence is, and it's like saliva, and what you do, this was the first bottleneck we took out of the COVID testing world, okay, was, wait, if they can spit in a tube and that's it, they can go in their bathroom, they can do this. You don't need a trained professional putting themselves at risk in PPE that was hard to come by, and 3-hour car lines, of drive-throughs are trying to get your sample taken. You just need a tube and you can spit in it. So that was the first bottleneck that we threw away. We said, forget that one, that's done. Yeah. The other thing that science is, and being myself, what we did with saliva when we first had it was, we got to stick this in the freezer, stick it in the -80. Right. It can't be stable. You're trying to measure viral RNA. There's no way it's stable. Stick it in the freezer. And we were, like, oh, we're going to have to use these buffers that, if we're shipping samples from here to there, we have to use a buffer to make sure that RNA is stable. And so there were a lot of these kits that were already, the one that was coming out of Rutgers, the Spectrum kit. Man, those are $18. They're not cheap. So if the kit costs that, we were defeated. Our whole pipeline was defeated. So we're like, no, there has to be, no, let's just test, just spit. That should be fine. Let's test it. Does it work? And we just started leaving positive samples out on the bench for like weeks on end. And every time we came back and tested it, it was like, oh, it's positive. It's positive. And we started putting it at high temperature. We started shipping it from, deserts. New York rates were going down. Right. New York rates were going down. Arizona and Nevada were going up, and we knew people out there. We connected with the hospitals. We're like, yeah, just ship it 110 degrees, whatever it is, just send it to us. Let's see how it happens. And it was still positive. So, and then the only thing we saw that actually destroyed the viral RNA in saliva was taking it out of the freezer and thawing it.
Oh man. That's the one thing that's gonna kill it.
It was so counterintuitive to everything that we had seen. And then Ann Wiley and Nathan Gruvot at Yale, I kid you not, instantaneously published something. Saliva, just spit, is so stable. You only need to spit in a tube. And it was like, oh my gosh, we are a match made in heaven. We need to be involved with each other. So
Right.
that's when we definitely teamed up with Yale. We wrote to them and, we were like, let's join forces here.
Yeah.
What are you doing? What can we do?
Yep. It's funny. There's the saying that, all others bring data, right? And one of my favorite lines that I've ever heard is way back at the first company that I ever worked for. There was a set of analysis, and the CEO refused to budge from previously held dogma, and the person that did the analysis was excellent in his own right, and he just deadpanned. He goes, "Well, don't let data stand in the way of your opinion," right? So
Yep.
it's something now where if you show it, you do it, you validate it.
Yes.
That's kind of where the trust comes from.
Yeah.
So now, the obvious thing to do was to send it to casinos in Las Vegas, correct? So I saw the headline that Wynn Casinos wanted to partner with you. So how did this come about, and where else have you sent the tests? And so
Right.
let's go to Las Vegas. How did this happen?
Well, so you would think a year or 2 years have gone by, right? But no, it's been literally, I don't know, 8 weeks, 10 weeks. It's been we finally got all the validation data that we needed. And we were speaking with the FDA, and the FDA said, you just need to submit this to the New York State Department of Health, if you want, as an LDT, and all this. And we're like, oh,
Yeah.
that's great. Thank you for the advice, and so we did. And it was like, yeah, go for it. Go ahead. You've done your IRB study. This looks good. Go ahead. Offer your service. You can commercialize, and we'll make some comments if we find anything that we find of an issue. So in August, we had enrolled a few schools. Because they were coming up with their reopening plans and they wanted to test, and I reached out to my kids' school and some other schools, and it was oh, this sounds great. Wait, you can do this for that cheap? Really? We can test everybody? And so we did that, and schools talk. So one school led to 10 schools, which led to 20 schools, which led to I think we're servicing school districts now. Now
these are public schools in New York.
So unfortunately, a lot of the public schools, have to follow suit all at once. And that's the hard part about it. So we had a lot of private schools jump on board. We've had our first public schools, join about a week, 2 weeks ago now. Actually, we were rolling out a pilot. Unfortunately, even though we've sent the information to both school districts and types and things like that, the public schools couldn't move as quickly, as a unit. So we actually had a PTA reach out to us. They said, to hell with our school, school won't take control. The PTA contacted
But we will.
us. Yes, we will. So the PTA, so we're working directly with the PTA. They organize it. They have the samples dropped off at school, before they go in.
Oh yeah.
And it's all PTA volunteers put this together. So, but we have, I think as far as Hawaii, universities, because they have NCAAs, all the sports, everything,
right.
So we probably service beyond, I don't know how many schools. It's, again, like I said, school districts now. So 15,000 weekly, 20,000 weekly from this, and it's a growing and growing exponentially, to the point where, and we have nursing homes, 31 locations across the country. We have sports, Olympic teams. The NBA, we're the NBA favorite, right? So all the tournaments, everything, we have to set up the plans of testing, turnaround time within, 12 to 24 hours, logistically, all of these things. So, I have been presenting in many places, and of course I'm from Las Vegas. And so, my brother's like, you got to tell Las Vegas, I need Las Vegas to be reopened. This is killing me. So, we have to present to the Nevada COVID task force, please, will you? And so my materials got sent there. I presented to them, and this is like, every single major casino CEO, decision maker is on this call that I'm presenting to. And they're just like, oh my goodness, what is this? And, we're just describing the pool testing, what it costs. And so Wynn reached out and said, okay, we do 300 weekly through UMC. on Fridays, 300 employees. We've been, the most aggressive, and we spot check our employees.
Yeah.
So they decided to do a side by side with us. And they sent the samples, and, we were 12 hours delayed in our results because of the shipping, right? It had to be shipped overnight, but the results were identical. And we did everything that we normally do, from pooled format, the narrowing it down. So that's what you asked me, how do you get to the individual?
Yeah.
So if we do pool in groups of 24, if it's negative, we consider everybody negative. If it's positive, we do what's called drilling down, which is called reflexing. So we'll go down and we'll give results of subpools of 2, because that's what New York State Department of Health allows us to do. They define a pool as any 2 specimens combined or more. So you can go down to that level without any physician order, without any personal information, without anything. And once you want to give an individual report, then you need all of those items. You need the physician order, right? So this allows you also to get around all of the regulatory and reporting administrative burden, right? You reserve the resources for those who need it at the very end. And for us, this has been very strategic, because our whole program is anonymous until you need that individual report. So only the last 2 do we ever get information on, and we never have to, actually, if they decide, we'll just send these last 2 to go get confirmatory tests by their physician.
Got it.
Right?
Got it.
So we may or may never have this information. And parents are extremely, thankful for the anonymity of the sample submission. My lab never only knows barcodes, and the school or the contractor is the holder of the key. Whose barcode belongs to which person. So that's how.
From a HIPAA perspective, you never get actual patient information.
Right. So we did this for the WIN, and the WIN was very satisfied with our results. And that's when, the CEO, Matt Maddox, reached out to me and said, I want this here, here, like in my casino. I need to reopen. What can we do? And I said, well, he said, let's talk to UMC. Let's see if they'll, run this lab. And I said, okay, if they'll run the lab, I can't run the lab there. I've got my own people, and I'm building it here. If they'll run the lab, let me go see what they're currently doing. If I believe in that we can easily retool their current pipeline quickly, then I have confidence that it will succeed. If it's something that they're not doing, and where it would fit into, it would be easy to retool, then I said, we're going to have a lot of hurdles. So let's see that. So it has been a wild ride, I have to say. It has been the fastest I've ever seen people move. We have an army of architects that are already breaking ground, building the lab right now. I mean, this was was, I don't know, what was this, like this was like beginning of September, no, like middle of September. I have no idea. It was very, September to October, we're breaking ground. We have all the blueprints building out right now. We have the regulatory being done. The team is UMC, WIN, and then Miramis team. So it's really going to be the WIN lab powered by Miramis's technology. And we're just creating this blueprint for them. And, what one casino wants, everyone wants. So,
Right. we can't not have that at our casino, because they have that at their casino.
Because they'll get all the patrons.
That's right. Well, then that's pretty fantastic. So you guys can potentially power all of Las Vegas's detection capabilities.
Yeah, so the goal right now is to build that blueprint, to build it successfully, show that this can work, that we can build a lab in 2 months and have this pipeline going. And if we can do that, we can hand this blueprint over and do this anywhere, right? So we have ideas for Boston, we have ideas for Florida. All of this is happening with partner labs. Miramis is not going to go and put its footprint in other places. Right. We're working with other labs that are already doing COVID testing and want to retool. Everyone always says, every time a new test comes out, I get all these headlines. Oh, the antigen test, this and this and this. Your test is going to be irrelevant when the antigen test come out. I'm like, yeah, no, not possible.
There's already hundreds of tests, right? Yeah. I mean, it seems like if anyone has paid attention over the last 7 months, it's not necessarily the test itself. It's how can people access it? How does the test actually get distributed? Yep. Right? Because the headlines and the test being published are fine, but who's actually using them, or who gets them? What do the actual results say? That seems to have been where the biggest blocks are.
we have had, good interactions with the NBA. The NBA has been comparing players, daily, asymptomatic players on a daily basis, antigen versus RT-PCR. And they have known and do not trust antigen tests whatsoever. They know that an antigen test can only pick up someone at a highly infectious state at about, 100 infectious units. That is, if you equate that into viral copies, it's roughly 10 to the 7 viral copies per ml. PCR can detect in the hundreds, 500 range copies per ml. You can detect someone at a pre-infectious state. They'll have a CT value roughly 31-ish, and that CT value can drop to 25 in 12 to 24 hours. If, and we know all of these things, from all of the combined data between Yale, between the NBA, between us and other groups that we're working with. So we have a collective weekly meeting with some of the heads of labs, running these studies all across the nation, to share these results. And so these are things we know. So the, antigen test is a test where you don't amplify, right? PCR, you're amplifying. So you're never going to get the sensitivity that you will. But, of course, they're going to have their value. So a part of our goals too is developing new technology. So we have a 30-minute test. It's working. It's pretty, fantastic. But again, the question is, how do you deploy it? How do you scale it? How do you make it actually work in the field? And this is where, having that audience at, the casino and having to build out, okay, they want to have people tested before going to a show. How do you combine the available testing strategies to make sure that you reduce the risk to, 1 in 5,000 chance that the person sitting next to you has COVID, where you have a higher chance of choking on your own food, right?
Right.
Oh,
This is
you wanted to use struck by lightning, but like the percentage wasn't high enough, so you had to go down the risk factor.
I'm working with people that like odds, right? They like to measure odds. So this is exactly the way they put it. This is what's going on, and it has been a really, daunting but fun challenge to get all of this going.
Prem, let me ask you a question about, let's go back and talk about timing from a practical standpoint, because you mentioned the potential 30-minute, test. So I'm a school parent, so I am in one of your schools. In fact, Chandra Ghost says, thank you. You apparently are doing their saliva testing for her child's school in Manhattan. So she thanks you. But so my child spits in the tube, and then I package it up and I drop it off where? At the school?
Yeah.
And then how long from the time you drop it off at the school till you either clear or have drilled down to figure out the individual positives?
So if you're local, we do have a courier service. They go and pick up, they have routes, so the moment it gets to our lab, everything is tested within 12 to 18 hours. It's tested, right? So, it'll get put into pools, it'll get, and if it's positive, we can watch it real time, and then we can take those original samples, right? So we take a small volume from each individual tube and then pool it. And the pool tube goes down the pipeline like any individual tube. So on any one given cycle that we do, we can test anywhere from 2,500 to 4,500 people, depending on the way we set it up in one PCR cycle.
Yes.
So what we do is we do that, and then we reserve, a little bit of room for if we have to do what's called the reflexing. So we just pull it out so that, so we're probably doing on the average of about, 3,000 people in one cycle. So, that takes, once you start setting it up, I mean, the reality, depending on, our staffing and all that, but it takes usually about 6 to 10 hours, once it's in the queue and going, right? So about 6 hours or so. You'll get the results usually in the middle of the night. If we usually say, okay, we deal with the results later. Because if it's negative, you'll get the results. If it's positive, we're calling people. That's an absolute, so they'll know right off the bat, we have a positive pool, and they'll know we're in the process of reflexing. So pools are usually, built around classrooms, things that make sense. You know,
that was my next question.
Yeah.
we got a lot of criticism because a lot of players were, oh, but if the prevalence rate outside is, really high, you can't do pooling. Well, no, you can, because you're creating a bubble. You're creating a testing
Right.
bubble by doing organizational based pooling. It's not the same as what you're doing outside, right? You're grouping kindergartners that interact with each other. So if you have a positive, you're likely going to have multiple in that class.
Right.
Right.
So we pool according to, the school kind of makes that, or the business does, all the people on the same floor, a restaurant, all the people that work, the same shift. That's the way it's done. So you get the results within 24 hours, usually your negative results. If it's positive, you usually get it quicker, and we're on the ball about it.
So I was thinking about this in the context of Las Vegas, but it could be really in the context of, anything. But, is there a chance to make it fast enough where, if you wanted to, go to a show in Las Vegas or go to a show on Broadway, if you had the samples in the morning or the day before, would you actually be able to clear somebody for that? Because ultimately, I think we're looking at really trying to figure out how things can open safely. And, is it possible to kind of stretch things fast enough where, should there be a Broadway lab, right? Where you go and you pick up your tickets, you drop off your test, and then at 6:30, they tell you to either, go to the show or not go to the show if you're positive.
Right. The problem with the PCR tests, right? We don't consider them rapid because they have to be brought back to a lab. There's some time, turnaround time, so if the lab is on site, yes, all of these things are feasible now, which is the goal, right? The problem with the rapid test is, how do you scale them? Even rapid PCRs, you do it one by one. I mean, how many machines do you need to get 3,000 done, right?
Right.
You still can't in a timely manner, even with a quick 10, 15-minute test. You do have to do them one by one. So there's problems with each of them. So you have to think strategically about, how do you pair them? What's more important? You have a show, and what's the time that you can expect someone to come and get tested. So maybe use a 30-minute, and then you're confirmatory. Everything is going to have to be confirmed by PCR. That's what we know.
Right.
So you can kind of give a high confidence of low risk with a quick test, but now you're going to confirm it in the back end, taking a little bit longer. So these are the strategies we're working out, right? We have the 30-minute test working. It's a color-based test. It's a little bit sensitive to pH, which the problem is we're using saliva. So if someone drinks orange juice, they're crap out of luck. They can't go to the test.
Right.
So we're retooling this to make it non-pH dependent, and then potentially try to pair that with the PCR backup, So these are things we're thinking about, so that depending on, if it's a convention, you have time, the 6-hour test is fine. That's a different story. If it's a show, and you're only going to be next to someone for a few hours anyway, we just need to make sure that for the next 4 hours, you were not infectious.
Contagious.
Right.
Right. So those are the questions you're asking are different. So you can design the testing strategy depending on, what it is you're doing, right? Like, having people test 72 hours before getting on a plane is totally useless. I mean, I don't even understand this concept. It makes people feel good, but it's totally, absolutely useless, right? So, I think the strategy of how you test and why you test and what do you expect to do with the results has to be more clearly defined.
Yeah. And I think there's part of the perception thing too, where you need people's perceptions to be reliant on data and actually good expectations, rather than, again, things that sound thorough or things that sound like a good idea because it sounds like you're doing more testing. We need to actually communicate the message where you can trust the results, and the results are going to be meaningful.
We actually have a question from the audience from Dave Hall. That's a nice wrap-up question, even though I don't think it was intended to be. It says, where will Miramis be in growth in 18 to 24 months after COVID has hopefully subsided? And what's the status of your fundraising to grow?
So, oddly enough, we've raised very little funding because we had a little bit of donations, and this has been sort of a fueling itself. Also, oddly enough, we have received $0 federal dollars on anything, considering my numerous conversations with the city, the state, everywhere, all over the place. And yet, who knows? I can't explain. But we've been able to successfully, roll this pipeline out regardless. And the capacity, I mean, we do have about, 50,000 people on our wait list right now for weekly testing. They're on our wait list.
Wow.
We are putting in extra, machinery, and we're hiring like madness to get them off the wait list. And we will accomplish that. That's not a problem at all. It was a matter of just making sure the equipment could come in, and we're doing that. So that will clear out. We will be at a capacity of probably, when we're running 24 hours a day, somewhere in the realm of 40,000 plus, individuals a day, right? We'll be doing more than states, whole states are doing. Cost-wise, I think I saw a question cost-wise. I'll tell you guys, we charge on a pool basis. It's $360 per pool, and this is for the school rating. This is the school cost. $360 for a pool, up to 24 people. So it roughly comes out to about $15 a person. For corporations, we have a lot of what we call feed forward. They're paying more because we're taking some of that and subsidizing the public schools. Right. So we have a foundation that we've spun out, and it's to help everybody have access. We have low income housing, we have all kinds of projects in play, and we're trying to feed forward into that. And so we have different levels of charging to help access for everybody. This is the plan. We're working with Testing for America, getting the HBCUs tested, on a weekly cadence and so forth. So it's grown crazily. Where we go from here, after people learn that they can spit in a tube, and you don't always have to have your blood drawn for things you want to measure. We can measure a lot of things through saliva, actually. Things we didn't know, things, and maybe this shifts the paradigm of what we can do, right, with saliva or anything. I think, again, when you can, in a matter of 6 months, take a concept, validate it, implement the study, and then commercialize it in 6 months, this pipeline, you can obviously figure out how to retool it for some useful purpose. So we have engagements with another healthcare company that is looking at that 18-month mark when we're not as crazy, into COVID, of what are we going to do next with the testing pipeline. And we're already starting and thinking about that and testing certain things along those lines. So, it doesn't worry me on, okay, COVID's gone. I think what we've been able to accomplish at the speed we have is just, incredible. The team that I have standing behind me has done such an amazing job. So the challenges will be presented to us, and we'll be able to, again, build a pipeline to accomplish that.
This has been amazingly inspiring. Now I'm going to right now contact our PTA president and see what our public schools can do. Prem, thank you so much for joining us. You are an amazing person, and the amount of creativity that you bring to such an important problem is really inspiring. So we thank you for spending time with us this morning. Thank you to TriNet for being our sponsor, and we look forward to your joining us next week as we talk all things election day. All right. Thanks, everyone.
Bye.
All right. Bye, everybody.
Bye.
Thanks, Prem.
Bye.
Thank you for tuning in to NewYorkBIO's virtual breakfast series. Join us every Tuesday at 9 a.m. for more discussions with leaders from across the healthcare spectrum. For more information on NewYorkBIO, please visit us at www.newyorkbio.org.